Scientists found that 2003 SARS survivors who have been vaccinated with the Pfizer-BioNTech mRNA vaccine produced highly potent functional antibodies that are capable of neutralizing not only all known SARS-CoV-2 variants of concern but also other animal coronaviruses that have the potential to cause human infection. The finding, published by Duke News Medical School and the National Center for Infectious Diseases in the New England Journal of Medicine, is the first time that such cross-neutralizing reactivity has been demonstrated in humans, and further boosts hopes of developing an effective and broad-spectrum next-generation vaccine against different coronaviruses. Among the coronavirus family, one subgroup relies on the ACE2 molecule to enter human cells. Both SARS-CoV-1 and SARS-CoV-2 belong to this group, as well as a number of coronaviruses circulating in animals such as bats, pangolins and civets. While the exact route of transmission remains unknown, these viruses have the potential to jump from animals to humans and could start the next pandemic. Collectively, this group of viruses is called sarbecavirus. We explored the possibility of inducing pan-sarbecavirus neutralizing antibodies that can block the common human ACE2 virus interaction, which will be protective not only against all known and unknown SARS-CoV-2 VOCs, but also future sarbecaviruses, said Dr. Chi-Hua Tan, Senior Research Fellow with Duke News Emerging Infectious Diseases Program and co-first author of this study. To test their hypothesis, researchers recruited 8 people who recovered from SARS-CoV-1, which was responsible for the 2003 SARS epidemic, as well as 10 healthy people and 10 COVID-19 survivors. They then compared the immune response of the 3 groups before and after they were vaccinated with the SARS-CoV-2 vaccine. In particular, they wanted to understand whether the neutralizing antibodies developed in SARS-vaccinated group could wipe out both SARS-CoV-1 and SARS-CoV-2 viruses as well as other sarbecaviruses, including potentially zoonotic sarbecaviruses that have been identified in bats and pangolins. Prior to vaccination, SARS-CoV-1 survivors had detectable neutralizing antibodies against SARS-CoV-1 but no or low-level anti-SARS-CoV-2 neutralizing antibodies. After receiving 2 doses of the mRNA vaccine, all displayed high levels of neutralizing antibodies against both SARS-CoV-1 and SARS-CoV-2, said Dr. Wani Chia, Research Fellow at Duke News Seed Program and co-first author of this study. Most importantly, they are the only group with a broad spectrum of neutralizing antibodies against 10 sarbecaviruses that were chosen to be examined. Our study points to a novel strategy for the development of next-generation vaccines, which will not only help us control the current COVID-19 pandemic, but may also prevent or reduce the risk of future pandemics caused by related viruses, said Professor Wang Linfa from Duke News Seed Program, who is the senior corresponding author of this study.